Welcome everybody to another REACH webinar. I'm Dr. Jacob Langer. I'm a pediatric surgeon in Toronto, and I'm on the board of REACH. And it's really a pleasure to be able to introduce Jenna today. She's a genetic counselor at NYU and is involved in the HDRC, the Hirschsprung Disease Research Collaborative, which is a long-standing study that we've been involved with Dr. Aravinda Chakravarti to try and sort out what the genetics of Hirschsprung disease are. We know that Hirschsprung disease is often runs in families and that there's a genetic basis. And there's been lots and lots of work done to try and sort out exactly what the inheritance is and how this affects what the what families who have a child with Hirschsprung disease, how they should manage this. So Jen's going to speak to us today for about half an hour to give us all the information we need, and then there should be lots of time left for questions afterwards. So Jenna, thank you for being here.
Thank you so much for that introduction, Dr. Langer. Can you hear me okay? Okay, awesome. I'll go ahead and share my screen. And you're able to see the slides? Great. Okay. Hi, everybody. It's such a pleasure to be here. As Dr. Langer mentioned, I'm a genetic counselor here at NYU Langone Health, and I have a dual role where I get to see patients with Hirschsprung disease in clinic, and then also, as he mentioned, I work in a genetics lab with Dr. Aravinda Chakravarti that conducts research. So a really big part of my role and my passion is bridging this gap between the research and the patient community and making sure everything we learn in the lab is actually communicated to patients. So I'm really grateful and excited to speak with you all today. I wanted to start with a brief bio because I know this audience is based all over the country and Canada, and I've lived quite a few places, so maybe we can connect over somewhere we have lived. But I was born and raised in the Bay Area in California, so San Francisco will always have a very special place in my heart. And I was lucky enough to go to New Orleans, my favorite city on earth, to complete my Bachelor of Science at Tulane University. And then I made the big move to the Northeast for graduate school. As you can imagine, I had to build my winter wardrobe from the ground up as I did not own a winter coat before then. But ultimately, that's what led me to NYU, where I still am today. So in the pediatric complex colorectal clinic, I see patients with all types of colorectal conditions, but 100% of the research I do at Dr. Chakravarti's lab is focused on Hirschsprung disease. So today we will start by brushing up on our general genetics knowledge, and then we'll briefly review Hirschsprung disease, even though you are all already experts on this. But then we'll combine those two and talk about what we know about how our genetics leads to this condition. And then we'll go over some genetic testing options available, and we should have plenty of time left over at the end for Q&A. But if you do have a question during the presentation, feel free to type it in the chat, and then we can, just so you don't forget it, and then we can read them all out at the end. Now, I love to bake, so I like to think of DNA as a recipe book. What do I mean by that? Well, recipes are filled with sets of instructions that make lots of different foods, and each recipe has many steps that you follow to make a finished product. There isn't just one recipe in a recipe book. There are hundreds. You might have a recipe for chocolate chip cookies, for lemon bars, for an apple pie, much more. Very similarly, our DNA contains many sets of instructions, but instead of creating food, they create us. And within our DNA, there are recipes that help create our fingers and our toes and our heart and, believe it or not, our GI tract. And another reason I really like this analogy is because every recipe is different. Your grandma's chocolate chip cookie recipe might be different from your best friend's. There might be some tweaks here and there, but they can still both be delicious. Similarly, there can be actual changes in the letters of our DNA between two people. Someone might have an A where someone else has a T. This kind of variation is completely normal, and it's what makes us all different and unique. We all have these differences in our DNA. Most of the time, these changes are completely harmless and they don't cause medical concerns, but they might influence things like height or eye color, or they might not do anything at all. But every once in a while, these genetic changes can cause severe medical concerns. Some people call these changes genetic mutations. I personally prefer the term genetic variants, and we'll use that for the rest of the presentation. So let's review. DNA is this long helical structure that contains the instructions or the recipes. It's like the physical pages of the recipe book. And by the way, pretty much every cell in our body contains our DNA, and our body is made up of trillions and trillions of cells. Genes are specific segments or sections of the DNA. So you can think of a gene like one step in the recipe, like add 1 teaspoon of salt. A variant, on the other hand, is now we're getting smaller. A variant is a specific change in the DNA code, like add 1 teaspoon of malt instead of salt. Malt is obviously very different from salt, and that would change the final product. And similarly, variants in a gene might change how it works and could ultimately affect how our cells function. So in red on the left, you can see in the sperm and the egg cell, you can see the DNA that's in the middle in the nucleus. And when sperm and egg come together during fertilization, there is now a full set of instructions and the recipe is complete. So now this fertilized egg can get to work. The cell divides and divides and divides again, and eventually forms a blob of cells. And during weeks 2 to 3, that's when fun stuff starts to happen. The cells start to migrate and differentiate, which is basically a fancy way of saying that the cells start moving around and changing shape until eventually they start forming our organs. Eventually, by week 4, there's already a little tube that will eventually form the GI tract. So throughout fetal development, these cells keep moving and changing shape until the full GI tract forms. And the GI tract is mostly by the end made up of different layers of muscle cells, as you can see in the image on the top right. But there's also some connective tissue cells, some immune cells, and also there's this network of nerve cells, which we call the enteric nervous system. So these nerve cells in the gut are essential for communication between your gut and your brain. The brain actually sends signals to the gut and tells it to contract and relax in order to pass stool. And we know that there are several genes, which I've listed on the bottom in red, that are really critical in driving this enteric nervous system formation. So these genes here are like critical steps in the recipe that forms the ENS. I want to point out in the top right these clusters of nerve cells, which are called ganglia, multiple ganglia or one ganglion. You might have heard this word before because Hirschsprung disease happens when our colon does not have these ganglion clusters and these nerve cells are not present, which is why Hirschsprung disease can also be called aganglionic megacolon. Aganglionic meaning no ganglion. I also want to point out, I know a lot of people use HD to abbreviate Hirschsprung disease. Our lab just likes to use HSCR since HD can stand for other diseases as well, like Huntington's disease and Hodgkin's disease. So just to not confuse them, we'll use that. I'll use that in the rest of this presentation. But anyways, as you guys all likely already know, Hirschsprung disease happens when the ganglia don't fully migrate down the GI tract during development. And as you can see in the image, the bottom part of the colon on the right does not have those yellow ganglia. The length of the affected segment can vary. So it can be short segment like this, but it can also extend longer segment. And sometimes it can encompass the entire colon. But without these nerve cells, that aganglionic segment of the colon can't communicate with the brain, and that tissue is just paralyzed, so it cannot contract and relax. This leads to functional obstruction, and then these babies cannot pass stool, leading to a backup of stool and an enlarged or distended colon, which is where the term megacolon comes from. And at that point, surgical intervention is necessary to remove the aganglionic segment. This is called a pull-through surgery. The incidence of Hirschsprung disease is 1 in 5,000 newborns, which sounds like a lot, but in the rare disease world, it's actually relatively common. There's also a male to female bias with this condition. About 4 times as many males have Hirschsprung disease compared to females, and we actually do not fully understand why this is at this point. Hirschsprung disease can also co-occur alongside other health problems, which I'll talk about a bit more on the next slide. But for most individuals with Hirschsprung disease, it's isolated, which means that it occurs alone with no other health problems. But for other individuals who have a genetic syndrome like Down syndrome, Hirschsprung disease is just one of many complications. So this is from our study cohort. You can see in this group of 981 participants with Hirschsprung disease, 58% had isolated Hirschsprung disease with no other health problems. But in 42% of cases, Hirschsprung disease co-occurred with other health problems, including other congenital anomalies like heart or kidney defects or cleft lip and palate, or also things like neurodevelopmental conditions, seizures, etc. We are currently in the process of preparing a manuscript that will summarize these findings and provide a clearer picture of the conditions most frequently seen alongside Hirschsprung disease. Among these individuals with additional health problems, though, some have a known genetic syndrome, such as Down syndrome, or Waardenburg syndrome type 4. Also, other commonly associated ones are Mowat-Wilson syndrome or congenital central hypoventilation syndrome, among others. But many individuals are not diagnosed with a known genetic syndrome. And a big reason for this is that the genetics of this condition are very complex. So sometimes genetic tests can't always detect the genetic cause of this condition. And I'll get into that a bit more later, and hopefully you'll have a better picture at the end of this talk as to why the genetics is so complex. So why does Hirschsprung disease happen? Why don't these nerve cells fully migrate down the colon in all of these individuals? The answer to this is our genetics. This just starts to show the complexity. But like we talked about before, there are variants or changes in our DNA code that can affect how our cells move and grow during development. So after years of research, Dr. Chakravarty specifically has been researching Hirschsprung disease for over 30 years, but the HDRC began in 2018. And after years of research, at least 24 different genes have been found to be associated with Hirschsprung disease, and there are likely many, many more. But the two main genes that I want to focus on are the RET, the RET gene, and the EDNRB gene. RET and EDNRB are both highly involved in enteric nervous system development. So it makes sense that when one of these are impaired or not working in any way, that this process of migration and differentiation will also be impaired, and the nerve cells won't fully migrate down the colon. RET and EDNRB don't work in a vacuum, though. They interact, as you see in this diagram, with a network of other genes. that all influence each other and work together to complete this process of enteric nervous system development. So what this means is that, let's say Rett doesn't have any variant and is working completely fine, but let's say RARB up here isn't working, then it is still possible that this can cause Hirschsprung disease because ultimately it still will affect Rett. Here's another analogy I like to use to really depict the complexity of this condition. Most genetic conditions that we hear about in the news, like sickle cell disease or cystic fibrosis, are caused by one gene. The gene is either working or it isn't. The light's either on or it's off. And if it's off, you get the disease. If it's on, you don't get the disease. These are called monogenic conditions. On the other hand, like we just mentioned, Hirschsprung disease can be caused by many genes. So rather than a simple on-off switch, it looks more like this dimmer light switch on the right. So we can think of our risk for developing Hirschsprung disease as more of a spectrum rather than a simple on-off switch. You can slowly dim the lights by moving different dials, and once you hit a certain threshold, you'll develop the disease. And I drew this scale with a threshold on the right side of the slide. An important takeaway from here is that not all variants are created equal. So some variants might have a small effect. You might be able to turn the knob a little bit for some of the genes, but other variants might have a much larger effect. So you can decide to move the dial a lot more, and that'll obviously cause the light to dim more. So let's just say you move the Rett dial down a pretty good amount, and that increases your risk threshold a pretty good amount. And then let's say you move the SEMA3C dial just a little bit to the left, and that brings you right over the threshold, and then you develop Hirschsprung disease. So that's how we think of Hirschsprung disease genetic risk. Alternatively, you can have multiple, like 5 to 10 of these smaller effect variants that can add up together and eventually meet that threshold. So that's why we call it a complex disease. So what do families with Hirschsprung disease actually look like? This is a family in our study cohort. And as you can see here, the first, let me point this out. Every letter that's in red is a genetic variant. So on the bottom row for everybody, that's going to be a variant with a larger effect. So anytime you see a red G, that's going to be a larger effect variant. Whereas anytime you see a red T on the top row, that's one of those smaller effect variants, so only moving the dial a little bit. So let's just focus on the left side. We see that in the first generation that this male individual does have a red G, so he has that rare coding variant, that larger effect variant, and he does not have Hirschsprung disease. His daughter, on the other hand, also has that same red G, that same larger effect variant, and she does have Hirschsprung disease, but it's short segment, but she does still have it. So just from this image, we can't explain why she meets the threshold and why he doesn't, but perhaps there's some other smaller variants that aren't represented in this diagram that just pushed her over the threshold and made her get it. But you can see that her partner has one of these smaller effect variants, this red T, and her partner doesn't have Hirschsprung disease, which we would expect because just that small variant wouldn't get him over the threshold. But now their daughter inherited both the smaller effect variant from the father as well as the larger effect variant from her mother, the red T and the red G. And you can see that she has a more severe form of Hirschsprung disease, long segment or total colonic. So that makes sense because she was likely brought over the threshold even more so than her mother was because of that extra small effect genetic variant. Now, this is another example. This is not a real family in our cohort, but just a example that I put together. But many families, if probably most families, look something like this that I've spoken to, where actually no one in the family has Hirschsprung disease until one child has Hirschsprung disease for the first time. So, how is this possible? Well, if we think back to that threshold effect, and let's think of this family as not having any of those larger effect variants, but only having those small effect variants. And let's say each asterisk here, it represents a small effect variant. And now let's set a hypothetical threshold and say that having five of these small effect variants will bring you to the threshold and have you develop Hirschsprung disease. Well, you can see that no one else in the family before the affected individual has five of these variants. They just have combinations of 0 to 4 of these small effect variants. And then for the first time, this male got or inherited five of these small effect variants, which brought him over the threshold, and which is why he developed Hirschsprung disease. Does that mean, now what does this mean for this individual's children? Will this male definitely pass down Hirschsprung disease to his future children? The answer is no, because he will not definitely pass down all five of those variants. He'll actually either pass down two or three. So it depends on how many of those small effect variants his reproductive partner has. And we actually do have specific percentages that represent this chance of passing down Hirschsprung disease to future generations, which is called recurrence risk. So in this individual, or I want to mention that recurrence risk varies depending on sex assigned at birth and also based on segment length. So in general, if you have a longer, more severe form of Hirschsprung disease, then you are more likely to pass down Hirschsprung disease than if you had short segment. So let's go back to the example. Let's pretend that this individual had short segment Hirschsprung disease. That will be in this column here. And because he is a male, he's an affected male, his children will actually only have a 0 to 1% chance. All of his males are 0 to 1, and any future daughters are less than 1% chance of passing down Hirschsprung disease based on this table. Let's say that his parents were to have more children, so his future siblings. That would mean, so any of his future brothers would have 4 to 5% chance of inheriting Hirschsprung disease, and any of his future sisters would have only 1% chance. Now let's pretend that this individual had long segment Hirschsprung disease. That would be this side of the table over here. And because he's an affected male, any of his children would actually, based on this table, have 12 to 19% chance of inheriting Hirschsprung, whereas any of his future siblings would have a 7 to 12% chance of inheriting Hirschsprung. So this table can be helpful when thinking about the chance of inheriting Hirschsprung disease, but notably, you can see on the bottom, this is a study from 1990, and all of these percentages were calculated based off of observational data, so empirical data, which is still very powerful, and this is a great resource. But at this time, we didn't have the genetic information on these families yet. So in the past few years, we have run whole genome sequencing on everybody in our cohort, and we're hoping to improve these estimates with our research in the future. Now, I wanted to transition to talking about genetic testing options available for you all. Genetic testing is not currently standard of care. So what that means is most clinics won't automatically refer you for genetic testing if you have Hirschsprung disease. A lot of times some clinics will, but for most of the time, if you are interested in genetic testing, you will likely have to advocate for yourself and reach out to your doctor and request to talk about genetic testing. I have listed a good resource here. If you're interested in looking for a genetic counselor in your area, you can go to findageneticcounselor.nsgc.org. I'll also put this link on the last slide. Um, so, so no need to scribble it down right now. But I also wanted to mention many of you might already be enrolled in our research study here at NYU, but if you are not, you are more than welcome, and you are interested, you're more than welcome to email hirschsprung@nyumc.org. This research study already has about 2,000 samples from 2,000 individuals with Hirschsprung disease. It's the largest genetic research study for Hirschsprung disease, definitely in the US, and I think the second largest in the world. And it's super important research for improving the testing and the calculations for recurrence risks, like I was talking about, and just counseling and management in general. Um, but back to clinical genetic testing. So if you decide to speak with a genetic counselor or your healthcare provider about clinical genetic testing, it's important to also discuss the pros and the cons of this type of testing. Starting with the pros, one benefit of getting clinical genetic testing is that you could get an answer for why you developed Hirschsprung disease. And some families like having that concrete answer and really feel that more information is power, and it's something they can point to. That said, only about 30 to 35% of people will test positive and will get that answer. And the reason for this is because clinical genetic tests actually don't test for those smaller effect variants. They only test for the larger effect variants. So they don't catch everything. Another potential benefit of genetic testing for Hirschsprung disease is management changes. So let's say that you test positive for a mutation in RET that is also associated with certain types of endocrine tumors. This is rare, but this is possible. Then you could get, you can meet with your oncologist and get increased screening and potentially preventative surgery, and you would have management changes for this genetic result. Another type of management change is family planning. So if you do test positive for one of these larger effect variants, there is a possibility for, depending on your state and insurance coverage varies per state, but there's the possibility for undergoing IVF with prenatal genetic testing to ensure that your future offspring does not inherit that same genetic mutation that caused the Hirschsprung disease. And in terms of potential cons or negatives of genetic testing, I already mentioned that clinical genetic testing doesn't test for everything. It misses those small effect variants. So about 70% of people will test negative. There's also a possibility of getting an uncertain result, which basically would mean that the lab found a genetic variant, but they're not quite sure as to whether it's the cause of the Hirschsprung disease or not. So some families might not like the idea of dealing with that uncertainty. Of course, cost and insurance is also a consideration, and genetic testing can be very expensive. But a lot of times insurance is catching up and will cover this type of testing for Hirschsprung disease. And then I wrote under other considerations because these can be benefits for some people or negatives for some people, depending on the person. But of course, genetic testing could have implications for other family members and could spark a conversation among family members about their risk. And depending on your family dynamics, this could be something that's stressful or maybe something that's empowering. And also, secondary findings is something you will discuss with your genetic counselor or healthcare provider at the time. But what that means is if you get a genetic test result that's unrelated to Hirschsprung disease and related to another health problem, in most genetic tests, have the option to opt in or opt out of those secondary findings. So in summary, there are many different genes, but primarily RET are responsible for the proper development of the enteric nervous system. Variants or changes in these genes can perturb this development, causing Hirschsprung disease. Hirschsprung disease genetics are complex. Different combinations of these smaller effect variants and these larger effect variants can add together to meet a certain disease threshold. Disease expression and severity is variable, even within families, and there are clinical genetic testing options and research opportunities available for you if you're interested. And I listed all those links again here. But thank you guys so much. And now we can open it up for questions for the rest of our time together.
Thank you so much, Jenna. That was a great summary, and I think hopefully people are much more clear on what exactly genes are and how variations in genes can result in Hirschsprung disease. We do have some questions. First one is that you mentioned that Hirschsprung disease can be associated with other health problems or syndromes. Can you go into a little bit more detail about what those are? And in particular, are they always evident at birth, or can they develop later? And how would you know if a child is at risk for one of these other conditions?
Yeah, that's a really good question. And that's a lot of what my research is focusing on right now. So hopefully I'll get a manuscript out about this soon that will be available to everybody. But most of previous research has focused on associated anomalies that are present at birth that are associated. So these are things like heart defects and kidney defects and cleft lip and palate and even structural brain anomalies. So a lot of the times these things are seen are said to be associated with Hirschsprung disease because they are found in those known genetic syndromes. So in Down syndrome, for example, we know that a lot of those kiddos have heart defects. So that will inflate that association between heart defects and Hirschsprung disease. But what my paper will be doing, it'll remove those individuals with Down syndrome and look at other individuals too with no known genetic syndrome. And we actually still see an association with these congenital anomalies, even when we're not talking about those known genetic syndromes. The reason for this is probably because of all that early embryologic development, those cells migrating that we were talking about. So those cells, they're called neural crest cells, and they migrate to form the enteric nervous system, but they also form some other parts. So sometimes genetic defects or genetic variants will cause both Hirschsprung disease and then also cause something else that's in that same pathway. Um, and then to answer your question, is it also only other things that are present at birth, or could it be health problems that are seen later in life? We do also see neurodevelopmental defects, or not defects, disorders, and things like seizures, and also some adult onset conditions, which I'll have to do more analysis on and get back to you.
Great, thank you. There's a question about research about ongoing issues that people with Hirschsprung disease can have, such as incontinence. Enterocolitis is another one that isn't mentioned in the question, but that we struggle with when we take care of people with Hirschsprung disease. Um, I guess because this is a genetics session, I would ask you, um, is there any way to predict these kinds of ongoing problems after surgery, based on the genetic variants that we've found?
That's an awesome question, especially because there's a PhD student in our lab looking at exactly that, and she defended her thesis this morning. Um, so I just watched a talk on this. But so the hypotheses for why some people have these long-term complications and others don't, there could be many different reasons. It could be the genetics, it could be surgical complications and maybe excess scar tissue or strictures or things like that, or maybe it's just random. Um, but her presentation really provides a compelling argument that genetics does have something to do with it. And more research needs to be done to say that for certain. But she shows that she shows that even looking at the colon later in life after birth, not just during development, that some of these genetic differences are you can see them. So yeah, great question. And the answer is we're looking into it.
Yeah, I mean, I would echo that as a person who takes care of children with Hirschsprung disease. It would be really helpful for us to be able to predict to some extent what kinds of problems people are going to have longer term and maybe change the way we manage those patients. So that's something for the future, but that is a great question. Um, there's a question, somebody says that her husband has Hirschsprung and so does her baby, but she was wondering if that means that she does she have genetic variants that aren't evident as Hirschsprung disease that she's passed down to her child. I think the answer is yes, but maybe you could just clarify that.
Yeah, so those smaller effect variants that we were talking about are really common. A lot of like I might have some we all might have some of these smaller effect variants. So it's very possible that you do have some of these smaller effect variants that didn't bring you to that threshold. Um, but especially if your son has a more severe version of Hirschsprung disease than your husband does, that could be a clue that that's possible, but it's certainly possible. Unfortunately, clinical genetic testing does not report those variants at this time. And the reason for that is because so many people would test positive for them, and there isn't exact management guidelines for who pretty much a lot of people, even people without Hirschsprung disease, would test positive. So that would just be lead to a lot of uncertainty, and it's difficult to interpret.
Thanks. There are several questions that are asking about if there's a relationship between how many people in a family have Hirschsprung and what the chances are for future children to have Hirschsprung.
That's that's a great question, and we don't have the exact percentages, but I can tell you just in general, the more people in the family that have Hirschsprung disease, that means there's probably more of those genetic variants that are bringing you to that threshold within the family. So the chance is higher. The more people in your family that have it, the more likely you are to pass it down.
Yeah, and there's also a question about the small variants that you're talking about and whether those can be tested for or whether it's just the large variants that can be tested for.
Yep. So at this time, the research lab, so the study that we do at NYU, the research testing can test for these small effect variants, but we can't actually report results because we are a research study. Um, the clinical genetic testing labs, they do not report these smaller variants because of what I was explaining earlier, because they're just so common. Um, Hopefully, though, on a brighter note, because I'm sure that many of you would like to know if you have these smaller effect variants. Hopefully in the future, once we have an exact threshold, because remember in that example, I set that 5 variants as an arbitrary hypothetical threshold. Once we have that number and know that it's exactly 5 or, something else, then at that point, labs would be more likely to actually report these variants to patients because then they would have exact management guidelines and they would know how to interpret it. So that just emphasizes the importance of research.
And just along those lines, with the you've given information for people to participate in research, but if they do that, will they get a report about what the results of that genetic testing is? I think that's an important clarification.
Yeah, so because we're a research study, we cannot report the results to patients, unfortunately.
So we have a genetic counselor on the call as well, and she was wondering whether you have an idea of where the research on Hirschsprung genetics is heading. Where do you think it's going?
Yeah, so I think a lot of these questions that have been asked is really where it's going. One topic will definitely be why, if we can ever predict who is going to have those post birth complications or post-surgical complications. I think refining the recurrence risks based off of our whole genome sequencing data is going to be an area of future research. So we got a grant from the NIH about 3 or 4 years ago where we were able to run whole genome sequencing on all of our patients, but we haven't finished analyzing that yet. So that's our next step is what can we do with this whole genome sequencing data? And one other thing we'll do is a lot of hopefully genotype phenotype correlation studies. So that's also a future area.
Great, thank you. That's very helpful. So uh, I'm just we have lots and lots of questions. So just for those of you that don't get your questions answered, I think what we'll do is maybe post the answers. Is that correct, Grace?
Yep, and we can also put our email in the chat. So if any of you want to reach out to us directly, we can get back to you through our email too.
Right. Okay. Well, there's one that's asking about medications and whether particular medications can increase the likelihood of Hirschsprung disease. I'm assuming that means medications taken by the mom while she's pregnant. Is there any interaction between those sorts of environmental things and the genetic variants?
Yeah, I mean, feel free to chime in also, Dr. Langer, on what you know. But what I know is that there are some studies out there about vitamin A, but they're ambiguous, and that there really isn't, according to Dr. Chakrabarty, any direct evidence about environmental factors.
Yeah, that's my understanding as well. So there are a few people that have been involved in the HDRC study over many years, and they've had additional children in the family who have Hirschsprung disease. Is there some way for them to update the information within the study?
Yes, definitely. You can reach out to either of these emails, and I would love to speak with you, and I'll update your chart.
Okay. There's a question about communicating genetic information after genetic testing uh, to to patients and to families. What are the guidelines in terms of how this is done?
Sorry, was it communicating specifically genetic information?
Yeah, genetic information.
Um, yeah, so because of the fact that these clinical genetic testing companies only report those larger effect variants, um, the guidelines are more clear because they're actionable. These are actionable results, and that we know are causative of the Hirschsprung disease. That said, this is kind of related, but maybe unrelated. But we're also doing a separate study right now where we are interviewing some participants and seeing some families of kids with Hirschsprung disease and seeing if they would want to know that information about the smaller effect variants. And we'll also just ask perspectives in general about how genetic risk information is communicated. So hopefully, we'll have better guidelines after that.
Yeah, I think, it's not just with Hirschsprung disease. I think, the whole area of genetic testing for all kinds of things has become much more complicated over the years. So these guidelines for when we should be doing genetic testing and how that information should be communicated is getting more complex.
Yes.
There's a question about this term mosaic variants. That somebody says that that their son has total intestinal Hirschsprung, and a mosaic variant was found in the father. Can you maybe explain what a mosaic variant is and how it should be interpreted?
Yeah. So I actually haven't heard of mosaic variants with Hirschsprung disease that often, but I'm sure they exist. But in general, so a mosaic variant would just mean that some of your cells carry that genetic variant, whereas some of your other cells do not. And that can happen rather than that variant being in the DNA and the sperm right away, so that all of your cells get the variant. Maybe that variant developed a little later in the progression, so then maybe half of your cells have the variant and half don't. So that's what a mosaic variant is. But in terms of its correlation with Hirschsprung disease, I'm not very familiar. I don't know if you've ever had a patient with a mosaic variant, Dr. Langer.
Not that I was aware of. Not for Hirschsprung disease. We do see it with other genetic conditions sometimes.
Yeah.
Um, Several people are wondering when the updated chart that shows the risks to siblings and the risks for children, when that might be available.
Yeah, that's a great question. I hope that now that we have the whole genome sequencing data, I hope that within the next 2 years, we can have better risk information available, recurrence risk information available. But for now, those risks are, from what I've seen, are not extremely accurate, but are pretty accurate. So I do think that's still a great resource to look at.
Yeah, I mean that's the resource that I have used in my practice as well. I guess what the question is, what do you do about it? And you know, and people wonder about prenatal testing for Hirschsprung variants and genes, mutations. Um, but I think it's important for people to understand that even if you found a variant in the fetus, that is present already in one of the parents, let's say, or in an aunt or uncle, that doesn't mean that that fetus will ultimately have Hirschsprung disease.
Mhm.
So, you have if you know that in advance, then some people might choose to terminate the pregnancy, for example, but that becomes very complicated because that fetus might not have developed Hirschsprung disease. So, do you have any further comments about that scenario in which people want to do fetal testing for Hirschsprung?
Yeah, definitely. I think if hopefully one of the major takeaways from this talk was convincing you all that knowing about these smaller effect variants matters and is important and can actually maybe inform some of those pregnancy decisions, because eventually, hopefully, we will know whether that fetus reaches that threshold or whether they don't. So I just need to convince the genetic testing companies that these small variants are important too.
Right. Um, has AI been used in the analysis? So you have so many patients now in the databases. Is AI being used to help with this research?
It is not right now. Yeah, it's not right now. We use RedCap database, but if there was a way to integrate AI with RedCap, I think that could be quite helpful. But we just haven't gotten there yet.
Um, what about um, what about using our understanding of these genetic variants to do gene editing or gene therapy? Is that something that you foresee sometime in the future?
Definitely. It's something that would happen once we like you mentioned, Dr. Langer, if we see a child with a variant, we don't know for sure that they will get Hirschsprung disease. So that's the main caveat. You probably wouldn't want to do any intervention like a gene editing if you don't know for sure that they will get Hirschsprung disease. But once we know more and we can our prediction models improve with this research, hopefully that will become a more likely option. That would be, I think, years down the line, but I definitely think it's a possibility in the future.
Great. There's a question about the effect of ethnicity or racial effects on the chance of developing Hirschsprung disease and whether there's an interaction between racial or ethnic groups and these variants that you're talking about.
Yeah, there's some epidemiological studies that in California that show that Black and African American populations have a 1 in 2,500 newborns develop Hirschsprung disease. So it's actually more common in certain populations than others. But in terms of the reason why, no research has looked at that yet.
What about, there's a question, can genetic testing help distinguish sporadic cases from familial cases?
Well, so just from knowing the family history alone, we would know even without genetic testing whether a case is sporadic or familial, because by definition, a familial case is when multiple people in the family have Hirschsprung disease. That said, the flip side of that, a familial family, let's say I'm testing a sporadic case and a familial case, the familial case is more likely to test positive than the sporadic case because it's more likely to have one of those larger effect variants.
Yeah, that's good. Because I think it's important. I mean, the definition of a familial case is somebody where there's a family member that has it, right?
Yeah.
Um, but it's the same variants that are causing Hirschsprung disease in the ones that do have a family history and the ones that don't. There's a question about whether the male-female difference is changing over time. Apparently, some of the Hirschsprung groups are finding more and more girls are affected. Is this something that you have seen in the studies?
This is not something that I've seen, but I will certainly look into that. Have you seen that?
I haven't noticed that in my practice, but an individual surgeon's practice is a relatively small number of
Right.
patients. And one thing that we have known for a long time is the short segment Hirschsprung population. It's 4-to-1 boys to girls, but in the long segment total colonic patients, it's about 50-50. And that's something that we've never quite understood because we do understand that the um, variants for total colonic Hirschsprungs and for short segment Hirschsprungs are the same variants.
Right.
And in a single family, I've certainly seen some people that have short segment and some people that have total colonic or even longer.
Right.
And presumably it's the same gene variants that are causing Hirschsprungs in all of them. So, do you have any sense of why there's a difference in total colonic in male to female distribution?
I mean, I can preface this by saying this is a complete guess based off of no data, but I would definitely think it's possible that it has something to do with those smaller effect variants, just because we haven't researched them as much. And yeah, I think it's possible.
I think we've covered most of them. There are some questions that are very specific that maybe um, we can that whoever's if anybody has not had their question answered, then you should contact us and we will answer it by email. Lots of comments saying that this was a great presentation and thanking you for your time and I would second that. I think this has been really helpful for people and it will be archived so people can watch it later on. And they can contact you. You've put your email on there so people can contact you directly as well.
Just thank you all so much. And yes, please feel free to reach out to me, the email on the bottom with any further questions. If you're interested in joining the research study or even just talking about the research study or asking more questions, I'm always happy to talk about genetics.
Right. And there were some there were some questions about how to find a genetic counselor near where they are, and I think you've got that. That's your first email on the slide here. So
yeah, this first link here is great. You can plug in your state and your city and look that way. If nothing comes up, just reach out to me and I can maybe also help.
Awesome. Thanks a lot, everybody.
Thank you all. Take care.
Bye-bye.